Hello learners, welcome back to my channel Be Curious. Today we gonna discuss very interesting topic, overview of the immune system. Let's start with definition. The immune system involved in organisms to defend against foreign pathogens like bacteria, parasites, fungi, viruses. It is a sophisticated system of defense that is capable of recognizing and distinguishing self from non-self. Immune system provides protection in two ways. First, a system of overlapping defense mechanisms which prevents infection. Second, an army of specialized cells and molecules that seek out and attack invading microbes. Most of the time, these protective mechanisms prevent infection. However, when infection does occur, damage to host tissue may result. This damage can be caused by microbes, their toxins, or by the immune response itself. Sometimes the immune response can damage healthy tissues, leading to allergic reactions or autoimmune diseases. In short, the immune system is a complex network that defends the body, maintains balance, and protects us from harmful invaders. To study of immunology, its numerous procedures and all the information we have about it has increased over the last few decades, perhaps thanks most to the study of infections and the study of immune deficiencies. This knowledge is crucial in understanding how the immune system works, how it can protect us, and how it can sometimes cause disease when it malfunctions. This chapter begins with a historical perspective on immunology, proceeds to discuss the fundamental concepts, and introduce the organs and cells that together form the foundation of our immune defense. Introduction. The following chapters explores challenging clinical situations where the immune system fails or becomes the aggressor. We cover how the immune system fights disease and many other medical aspects of immunology in detail. A historical perspective of immunology. Immunology began with the observation that people who had recovered from certain infectious diseases were protected from the same disease again. The Latin term immunis means exempt. In English, immunity means protection from infection. In 430 BC, Thucydides wrote in Athens that people who recovered from the plague were less likely to fall sick again if they caused it a second time. 18th century. In 1796, Dr. Edward Jenner noticed that milkmaids who had cowpox did not get smallpox. He inoculated people with cowpox material and showed it protected them from smallpox. Jenner techniques. In this technique, cowpox from cow inoculate into a healthy person, then found it protected from smallpox. Early vaccination studies led the way to immunology. Early attempts to induce immunity date back to Chinese and Turkish practices 10th century to prevent smallpox. By the 1700s, efforts were made to prevent smallpox using material from smallpox pustules. Lady Mary Wortley Montagu in 1718, after seeing this in Constantinople, introduced the practice in England. Edward Jenner in 1796 used cowpox to protect against smallpox, a major breakthrough. Jenner's idea spread quickly across Europe and laid the foundation for modern vaccines. In this picture, showing that African child with rash typical of smallpox on face, chest and arms. The cowpox story, Jenner discovery. My milkmaid who got cowpox, a mild disease from cow, did not get smallpox. Jenner collected material from cowpox pustules and inoculated it into a young boy. The boy developed mild cowpox but did not get smallpox when exposed to it later. Lack of fresh cases led Jenner to inoculate older children and later even chickens, showing protection across species. Key takeaway: immunity has been observed since ancient times. Vaccination discoveries in the 18th century opened the door to modern immunology. Observation, curiosity, experimentation is equal to protection against disease. Vaccinia, the key to vaccination. Vaccinia, from the Latin vacca means cow, is a word that was derived from cowpox. Postulates caused from fluid taken from cowpox lesions could protect people from smallpox. This idea was tested by Edward Jenner in 1796. In 1881, Pasteur first conducted an experiment with chickens. He found, as Jenner had suspected, that people who were exposed to weakened microbes were protected from the disease. In the 20th century, Jonas Salk and Albert Sabin developed the vaccines against polio, transforming the disease from a dreaded illness to a largely preventable one. Today, we still build on the principle of Jenner, Pasteur, and others. Key idea: the idea that exposure to a weakened form of a microbe can protect against the disease is the foundation of modern vaccination. Jenner in 1796 used cowpox to protect against smallpox. Pasteur in 1881 proved that weakened microbe can protect. In 20th century, Salt and Sabin developed polio vaccines. Vaccination in organs. Vaccination for stronger immunity. The concept of loosely attenuating smallpox and other viruses to give slightly weaker but immunogenic, able to stimulate immunity strains to host organism was a crucial process dating back to the 18th century. And as early as the first decade of the century, this process also used with the organ systems of mammals. Different organs have a special capacity to resist or adapt to the effects of vaccines. This adaptation provides long-lasting immunity against various diseases. How organ vaccination works. A weakened attenuated microbe or its components are introduced into an organ. The organ cells and immune components recognize the microbe as foreign. They activate immune cells, B cells, T cells, macrophages. These activated cells produce antibodies and memory cells. When the real dangerous microbe enters, later the body responds quickly and prevents disease. Example of organ used for vaccination: skin, lungs, intestines, lymph nodes, spleen, bone marrow. Key takeaway: vaccination began with observation and simple experiments. Weakened microbes equal to protection without causing the disease. Different organs help build long lasting and strong immunity. The next major advance: Louis Pasteur. Louis Pasteur in 1822 to 1895 proved that microbes cause disease and developed the principle of attenuation to create vaccines. Pasteur cholera experiment. In this experiment, Pasteur grew the bacteria cholera in the broth and heated the culture, then injected into chicken and found that chickens remained healthy. Pasteur attenuation experiment. In this experiment, Pasteur cultured the bacteria cholera, then kept the culture for weeks, months in air, not fresh broth. Then bacteria became weaker, attenuated, not able to cause strong disease, injected into chickens and found that chickens stay healthy. Next step: later, when injected with a strong and virulent cholera bacteria, the chickens were still protected. This showed that weakened attenuated microbes can train the immune system and provide protection against the real disease. Why this was a game changer? Proved that microbes cause disease, showed that weakened form can protect, laid the foundation for modern vaccine, rabies and anthrax. From ancient observation to modern vaccine, the quest to understand immunity has saved millions of lives. Return to fresh chicken and control. Sometimes after a summer vacation, Pasteur returned to his lab. He found his old bacterial culture still alive. Then he injected these fresh, virulent, strong bacteria into chickens. The chickens died. This confirmed it was the live, virulent bacteria that caused the disease, not the air or other factors. Attenuation, the key idea. Virulence, disease causing power of microbes, can be weakened by growing them in old culture, exposing them to air, changing temperature, other environmental conditions. This is called attenuation. Attenuated microbes safe but can still protect. In summary, observation, then curiosity, then experimentation, then discovery. The path of science. Pasteur's work showed that attenuation can induce immunity without causing the disease. This principle is the basis of many live attenuated vaccines used today, like BCG, MMR, oral polio vaccine.